This is a short summary video on DNA profiling and specifically PCR. So I'm going to make this as quickly as I can. The point of PCR is to adequately amplify the DNA sample. So for example, if you're at a crime scene and you have 5 cells, for example, I know that sounds like a very small number, but if you had 5 cells, you could use this technique to amplify it to have hundreds of thousands of DNA, of the same DNA sample in a very short amount of time. How does this take place? Okay, so you need 4 main things. Just remember, you need 4 main things. You need the DNA sample in question. You need DNA polymerase, which will catalyze the reaction of the addition of free nucleotides to both strands of the initial DNA sample. You need a primer to make each single DNA strand a double strand, because DNA polymerase only works on double-stranded nucleotides. And you also need the free nucleotides. Okay, so the key to this is that the purple line are the DNA samples, the primers are in red, and the free nucleotides are in green. Let's run through the method. Now, at first you have your DNA sample. Now what you want to do is you want to denature that and heat it to 96 degrees. This breaks all the hydrogen bonds between the anti-parallel double-stranded double helix. Double-stranded double helix. And then once you have that and the strands are separated, what you'll have is a cooling to 68 degrees, and this allows the primers to anneal. And have I completely skipped that? Okay, no, they denature, you cool them, you have the primers annealing at both ends, the 5 to 3 prime end and 3 to 5 prime end. Then you heat it to 72 degrees because that is the optimum temperature of Taq polymerase. They bind on. The DNA polymerase can then bind, and it can begin to add free nucleotides to each strand, and lo and behold, you have 2 new strands. And then you can simply bump up the temperature to denature it again and take it from the top to the bottom. You can repeat this process and it begins to multiply exponentially. So it goes from 2, then it'll go to 4, then to 8, then to 16, etc., etc. And that is pretty much PCR.